We start, sir. So, very good afternoon to everyone who's joined us with us for this webinar. This afternoon, it's my privilege and honor to welcome Dr. Rajpal Singh Kashyap, who's the director and senior scientist at CIIMS Advanced Research Center at Nagpur. Sir is a very well-known person to everyone, mostly in Nagpur, and now we are also. It's nice to get to know him. He's a very vibrant, dynamic person, and is fully enthusiastic in research, not just data, but also a lot of community work, and he's also a part of multiple projects which are actually shaping national policy. And also, the most interesting thing about him is the wide spectrum of research he does. Not just diagnostics, it also involves management, it also involves community exposures, and so on and so forth. So it's very interesting to hear from him today on insights into the brain infections from experience from the Central India. And we hope through this webinar, this is an educational initiative of our Center for Advanced Research project, which is focusing on the impact of climate change and brain infections. And through this, this is an educational initiative across the nation where people hear from different experts and enhance their knowledge on various brain infections. Thank you, sir, for your kind acceptance, and over to you for the webinar.
Am I audible, Angel?
Yes, sir, you're audible. Yeah.
All right. So thank you, Angel, and thank you, everyone, for your, time. That's the most precious thing. And I don't know, the things which we are going to discuss today is a little bit about our institute thing, because I want to share, because this is almost a 45-year-old institute. So I thought I should share my experience and something related to project, something related to what we have experienced, and some of the things which we can do in future also, and some of the issues actually which are really we don't understand till now. So maybe going to talk on some aspects in next 30-40 minutes. It depends on you, how much you listen, how much, because I was talking to Angel a little bit earlier that speaking is, I can speak 2 hours, but listening is very difficult. And especially listening through this online mode is again difficult, and difficult for speaker also. So coming to your thing, so I'll switch over to the next slide. Right, so a little bit about institute. As I have already indicated, it's a neurological institute, and it is the first institute in Nagpur, and it was established through Dr. G.M. Tavri. And as I have already float a lot of information and my things in the social media also, when we have collaboration with CMC Vellore. So we all remember Dr. Tavri because he is from CMC Vellore. And when we check the file and all these things, the angel sent to everyone, and I can see the mirror image of everything. So it looks like to be the same thing what he has done, what he was doing in CMC Vellore. So this is how this institute is established, and now we have so many neurological institute in this part of world, and most of the people worked here, and then they have started their own hospital. So this is a parent institute for Central India, this thing. And when we talk about tertiary care center, so I think it's we all understand the type of cases we get in tertiary care. So it's really important for us to understand we don't get straightforward cases. The people come here with so many diseases. They don't tell us good history. We don't know how many times they have taken treatment, where they have gone, what kind of treatment they have taken. So absolutely not aware of the basically the thing about the diagnosis and all these things. And taking history is let me tell you, it's people don't tell you correct history. That's the problem. We understand it that they are telling us their history and all these things, but it is really difficult. So few things about before I go about my view about my institute. The institution profile already indicated it is in Nagpur, which is a 0 mile center for everyone. We have 0 mile here. So this is the place, and this is a leading tertiary care center. And most of the population we get from Madhya Pradesh and Chhattisgarh, and some of the border side of Hyderabad to Maharashtra side. So we are getting all these things, and most active department is neuroscience department. We do have other facility because now whatever required for all these things, so we do have other facility, but primarily the majority is basically neuroscience department. And I always say when I go to ICMR, to policy making places, PMO and all these things, I said, what to do with this, the data, the clinical data and clinical samples we have, right? I mean, it's very painful when you're throwing all these samples, what to do with these samples? So I'm just requesting ICMR and all other scientific agencies and PMOs have a clinical metadata thing because we are losing the data. Not only in neurosciences, in every clinical angle, we are losing the data. I mean, in cancer, it's a real-time data. What type of cancers, what populations are involved, what kind of disease they have. We actually not aware of it. Once clinicians see those data, and then we forget. That's all. So I think there is a need for a basic scientist, researchers, clinicians to come together. There is a need for centers where we have this kind of a national data where we can get all this kind of a this thing and biological samples. When I go to clinical, conferences, basic where people are presenting their basic data, they're struggling for clinical samples, and we are throwing it. So I think it's a gap which we have to figure it out, and this is our strength basically, and that is what I think. The best part is this institute, like what I have seen in CMC, the clinician and scientist integration is very important here. It's all because of Dr. Tavri. He has that kind of a, he given us that kind of a platform where clinicians are also interested. Generally in India, clinicians are not able to do research because they have a lot of things to do. I've seen people looking onto 100 patients per day, 90 patients per day. So, and then follow up rounds and so many things. So it's very difficult to work on the research questions, and in-depth research is really difficult. So I think this kind of integration, what we are doing at the moment, every week they come down to us, and then on the table we discuss. They come with a problem, Rajpal. These are the issues we have. These are the things can be done. So this is what we gather from bench and will deliver basically from bedside and deliver to bench, and bench revert back to them. So basically it's all about bed to bench and bench to bed. This is what exactly we are working on these things. And the important thing is what I talking about the data, the clinical samples is longitudinal approach. Right. I'll share some of the slides to you when I talk about neuroinflammation and all these things. So but how follow-up samples are important. And I think follow-up samples are really important than the original samples. Of course, the second samples give you more information than the first samples because it characterizes the first samples. It rectifies the first samples, whether we are on the streamline or what kind of changes we have seen in all these things, whether it's a CSF profile, whether it's a follow-up of any infectious disease, or clinical metadata or not, whatnot. We can find out with all these things. So this is what exactly the strength and the research philosophy of Institute. And no doubt about that, the policy making already indicated the bedside to bench approach. Idea is, if we can start things at our institute, that is, I think, we have done our job. I mean, we cannot go to the global level immediately, right? Because you have to work in your periphery, you have to work in central India, you have to work in a Nagpur region, and whatever the things. There are several examples which I can tell, which actually research tells clinicians, now you have to revise these things in this way, and the policy changes, right? Then bombardment of treatment changes. So basically, this is really useful that it should be used in your institute. Coming to the topic, neuroinfections, we all know, I don't have to tell you, when it goes to the brain, the bugs goes to the brain, the complication is 300 times more difficult than in other infections you have and all these things. And now, since I'm working on AMR, I have seen so many difficult things. We have seen patients not responding to the treatment. It's really painful at the bedside. So basically, AMR is definitely a problem. Yesterday we had a meeting that hospital acquired infections from a ICMR and from other policymakers. They were in the meeting and we had discussed about all these things. So it's a very difficult thing to handle AMR and all these things, because when you talk about bugs, believe me, the bugs were there when we don't exist, and they will remain there when we don't exist again. So bugs will never go anywhere. They were here, but we had to manage in a different way that they should be not harmful to us. So you cannot change everything. The issue is we are very ambitious when we talk about EMR, when we talk about, so what we feel that it's going to be change everything when I do research. The research will hint you. Research will just give you a clue. Then you have to think about at the local level. And I completely, confident that type of cases which we are looking at our CIIMS, it might be different at CMC Vellore, the approach will be different and the conditions will be different, host will be different. So I think there are different things as to be there. So I think if you work on a particular patient through the angle of research, what you have done, then it's actually research. That's the actual thing you are translating. But you cannot take data, you understand from research. That's all I can say about research. Research will hint only. That's the underlying thing. I just want to tell all to the listeners, research will never give you answer. Will never give you answer. I've seen people who worked on 40 years, 45 years. So it is no answer. If you ask me, sir, what is the simple answer? I don't have any answer. After doing 30 years of research, I don't have any answer. So this is really important when you talk about EMR and all these things. Another thing is BBB, right? Drugs don't go to the brain. If you give 100 mg, how much it reaches to the brain, it all depends on the blood-brain barrier integrity of that particular patient. Whether, we are working on the climate change also. So whether climate change affecting all these things, we don't know. So this is what exactly we don't know. And people have reported with in detail pharmacokinetics and all pharmacology studies, the drug don't go into the brain the amount we give. So these are the issues we have, and these are the challenges we have. Diagnosis is one thing, penetration, what I have talked about, all these things. So not taking much time on this aspect, I can switch over to first part, what we are doing at the moment. And by giving examples of our, we can definitely move towards the other thing which we have done in these things. So diagnosis, we all know when we talk about brain TB, very difficult, right? Because you have a few number of bugs in the CSF. It's a very sterile area. So few number of bugs in this thing. So difficult to culture, difficult to stain and all these things sometimes, especially when you talk about TB, right? You need an immediate treatment. But the thing is, AFB is almost 90% of the time it's negative. right? And culture takes, even you put it in the liquid culture or in advanced culture system at the moment we have, it takes more time. I mean, clinician can't wait for treatment for this particular thing. And then if you remember, if you can go and see one paper, Guy Thwaites have written about different way of staining of AFB, and he has given some advanced way of AFB staining. Look into different field. Microbiologists may be aware of that thing, and then the positivity of AFB is increased. So everybody is trying their best level to understand whether the bugs are there or not. And then culture is another limited thing. We know gold standard, we don't get it, and CSF picture, it's always a problem when people are already treated. I mean, they come with a different type of picture when it comes to you. So ultimately, what we have done, we switch over to PCR things in our institute. And initially, we have started with this 8 bucks. This is what our clinicians have given to us, that why don't we work on these things. And this panel is already established in our institute. So 8 pathogens usually we do in our institute. These are the report format. Now we move to the combo panel. I'll come to the virus also. And these are the things we do, including this this thing, and then the combo. It all depends on the clinicians, what they say. Now, to using this molecular biology things, we are able to diagnose better than what we done with this staining, culture, and other method. The positivity is quite good, and we started getting some kind of a samples. But still, diagnosis is really an issue. You believe me or not, I mean, you can send it to 4 places and see the results, right? People don't do it. I mean, send it 6 places and see the result. I mean, nothing is matching. Nothing is matching. That is the most difficult thing in the diagnosis, and the very afraid thing, what I think, because I have sent same CSF sample for CSF biochemical things, for CSF profile, TLC, DLC, protein, sugar. See, I have data of 300 patients, which is not matching. Now, what to do? The clinician generally see it's a low sugar thing and it's a lymphocytic picture or something like that. They start with either it's a brain TB or other bacterial meningitis or pyogenic meningitis, something like that. So it's a really difficult task. Diagnosis is the first step, where we are still, from so many years, we are struggling. And it is a thing which we are, despite of so many advanced technology, uh, the only challenge I'll give to everyone, do it in a 6 place and then come with the results, and do it in 100 samples. You'll come to know how the diagnosis works. So that's the difficult thing. The ultimately response to treatment and all these things have to be figured it out. And the follow-up samples, as I have indicated, a continuous positivity of that particular bug is really important. And I think that's the thing which we have to figure it out. So this is what exactly this improved good positivity we got with this testing and all these things. So we reached to this level. And similarly, we have antibody detections. And in the previous slide, I have shown PCR also. When we talk about VZV, Japanese encephalitis, entero, adeno, and we are doing Chandipura also, since we got project from these things. And not only this thing, we are approaching towards multiple approach. You don't know at what stage the patient comes to you, right? So maybe at the early stage, you may get some antigenic load. Later on, you can get antibody, IgM, IgG, and then molecular assays and all. So it's a better thing. What I suggest in our laboratory, it's better to do everything, right? Maybe a cost effective, it's not a cost effective thing, but it's not costly also, because it's a very simple test and you can do it at different stages. So even we have data where we have shown that the antigen is negative and antibody is positive, IgM is negative, or vice versa. So this type of results you do see actually on the bedside thing, right? You don't get a neat and clean graphs, right? When you're working on the clinical side, when you're working on actually on the bench, you don't get anything very clean, like we show in the manuscripts and all these things. We filter it and then we clean the data and then publish it. Otherwise, at the bedside level, we get a very complicated thing and very complicated kind of diagnosis and all these things. But we are trying our level best. like all these things. My team is working on all. Even now, we switch over to antipeptides also, because peptides, antipeptides diagnosis is quite very easy to do, and you can generate peptides, signatures, several signatures. So this will give you more confidence, right? If you use a 20 kD protein and a different sites of peptides, we can use it and use antipeptides for that thing. The chances of getting positivity is quite high, and initially it's costly when you prepare, but in a long term you get in 10 mg of peptides, 10 mg of antipeptides. So it works for so many years if it's stored in a proper liquids and all these things. So these are the few reports which we are doing it in our institute. There are some supporting tests we tried in our institute. ADA is the thing. I think this two tests actually, ADA is non-specific. We all know it's a T cell marker, and activity shows activity of T cells. So it is not very accurate, but it is useful in prognosis, right? If you have a definite case of TBM, and if you need a follow-up samples, you don't want to do anything, ADA will give you some kind of indication. Quite a supporting test is that. Two places I have seen ADA is reliable. We have published 2-3 paper on ADA in the BMC infectious diseases and complementary, I don't know, CSF research. Sorry, this is the paper actually we have published long back, and the two areas where we find ADA really very significant. One in CSF, the other is in pleural TB. There is something which we don't know what we are getting out of this thing, but ADA seems to be a good thing. And we tried with our IIT partners and all these things, whether this can be done, develop at the chip-based things. We are trying our best level. Somehow we are not able to work it out, but we are preparing in a way, because it costs 50 rupees, 40 rupees, 30 rupees. So we just want to try it at technical level, whether this can be used at a routine screening or something like that, or it will tell you some infection. So not succeed at the moment, but still trying our level best and all these things. So this is all about the bacterial meningitis and viral meningitis diagnosis we are doing in our institute. And now I'm moving towards little bit other things which we are doing is zoonotic infection. So, I mean, and now, institute, we have ICMR Institute in Nagpur, National Institute of One Health. So this was basically originated from this data. Actually, we have started working with ICMR around 12 years back. And then we have gathered around 8,000, more than 8,000 human samples in 5 years. And there were 6 institutes were involved. 3 is there and 3 senatorial centers were there. So 6 institutes were involved, and then you can, with simply 10, 12 crore they have given to us, and then we have figured it out and then find first time the zoonotic infections in this part of the region. Generally, even clinicians are not initially ready to do this kind of a test, but when we saw 6.4, 11, 1.5% positivity in all these things, then we got these things. The interesting fact which I want to share with you, we have done around 200, 300 samples where we have some positivity with Listeria meningitis cases also. So this is generally we miss it. We don't focus on this kind of zoonotic infections. Maybe the test we have used or the primer we have used, honestly speaking, 10 years back, if I come with a better diagnosis today, the positivity may be very high. And possibly the positivity is high. Some of these strains do cross the blood-brain barrier and cause Listeria meningitis. We do got bovine strain positivity, bovine TB meningitis in CSF also using these things, and brucella, neurobrucellosis. So this is what exactly we have reported. And interestingly, we are doing routinely in our institute also. So this earlier it is neglected diseases. Even at some places, people are still not doing for simple brucellosis and Listeria and all these things. And now in Nagpur, we have very good One Health team, Environmental Institute, NEERI, you are aware of it. It is taking care of environmental thing. and MAPSU is for animal health, and CIIMS and other centers are there for this clinical part and all these things. So this is what exactly we have reported, and this is all from Madhya Pradesh, Chhattisgarh, and all these places we have seen. This is about the One Health perspective, because Angel was interested that I should talk on a little bit on the One Health aspect. So let me tell you, I mean, when you talk about neuroinfections, so this is actually the last thing which you see after infection or all these things, because you get infection and then it maybe go to the crossing the blood-brain barrier and cause, like what we have seen in neurobrucellosis, listeria, bovine, and we have reported scrub typhus in CSF also. So these are the bugs which we have reported, and definitely this is really important. We got one intermediate project of around 9.5 CR, where again this One Health team is working, and not only the human animal concept, but we have environment things also. We are included all these things and doing NGS of around 200 plus samples of this stool samples from human, and then we have stool samples from animal, then sorry, fecal from animal, and milk from animal, then soil, wastewater, drinking water. So we are looking all these things with a broad perspective. What is the transmission? What are the genes? And what are the things we can find it out from? Actually, we all are blaming one to other person, that it's coming from animal, it's going to humans or what. So this is what exactly will come into picture. We'll try to understand little bit all these things, and then some of the cases having some CNS thing and all these things. So we are looking at the transmission pathways, and these are the teams which actually, it's a ICMR project. And Urmi, if you can see right side of the screen, now she is in University of Delhi, South Campus. She is responsible for having bacteriophages thing. And she worked on the mycobacteriophages also. So she prepared bacteriophages, and it's good to see that Dr. Kurkure of Animal Health is using all these things in a field for his some of the animals and all these things. So I don't have right to share those data at the moment because it's still in the research. But bacteriophages might working better and may give us some help. I don't say it will eliminate the AMR responsible AMR things and all these things, but I think it seems a little bit supportive for bacteriophages if we can work more on this aspect. And this is what we are doing, basically MDR pathogens and all these things. So this I just want to share for students if they are there. This is the plan which we are collecting and then getting samples from One Health perspective. So I think we need to add neuroinfection also in One Health angle. It's a time that we have to see from to the lower thing to the higher thing. What are the things are involvement? Because yesterday somebody was talking in the hospital associated infection. It was like controlling infection in the hospital is another big project, right? Where you can spend crores of money because you have to do a lot of things. You have to change curtains. You have to have good people for cleaning and all these things. So, but actually these are responsible. These are the small things, but have bigger impact. So I think we need to involve this kind of approach in neuroinfection. Maybe right now at the moment we are working at the climate change. So climate change is nothing. It's coming from an environment thing. So ultimately, when we have to show practical things, right? For example, you have to show this environment showing this thing and that these bugs are there. Let's work on the ground level also, how it's happening, and then this kind of a thing. This is what we are doing it with this thing. What kind of a heat map we are getting from different places from different time. You can see it's from animal feed, drinking water, soil, water, different kind of samples we are taking, and different farmhouse in the sense where they have a lot of animals, and this is what we are looking. And also the AMR heat map also I've shown in the screen, where you are looking at different kinds of bugs in human samples. So this was about, I'm just talking in pockets, one after another, one area, just kind of a thing so that we can work future in these things. Now moving towards the undiagnosed meningoencephalitis infection. Now, I mean, we all, those who are working in neurology, they are aware of it. I have already indicated, very difficult to diagnose. And still clinicians believe the combo panel and the system we have diagnosis or anybody have, still we are missing 30 to 40% or 50% cases. And it's always disconcordance with the clinicians, right? Because they clinically see, they see very clearly, and they see response to the response to the treatment. And this is what we have to actually do it. I don't know whether the 4-AKT, when we do for TB, the 4 anti-TB treatment healing something else also. We don't know that. But when it responds to anti-TB medication, we call it TBM, brain TB. Similarly, there are a lot of meningoencephalitis infection. So what we have done, we have written to two places for this project. One is BMGF. It's a Bill and Melinda Gates Foundation, and Indian Council of Medical Research, and then CZID. Basically, it's a metagenomic institute in San Francisco. So they have, the test tube, the number of test tube we have in our lab, they have all NGS instrument in their lab. So they have hundreds of NGS, bigger, smaller, and smaller. So what not? Everything they have for NGS. So I was there with my team there for 15-20 days to studying whether this metagenomic approach, shotgun metagenomic approach, can be useful to diagnose some cases and find out the cases which we are not able to find out through PCR, whether we'll able to find out through this kind of a thing. So we worked on it. I don't want to take it, stretch it, because this is BioFire, all this thing, the approach we have done. The first thing what we have done, because if you remember last time I was talking to Angel, when you go for a metagenomics, you have to take care of it, everything. Even a small contamination in the environment create the problem. If you don't have a proper control samples, if you don't have a blank samples proper for NGS, this will spoil all your testing and everything. So this is very important to get negative thing in the negative cases. So basically, we worked on the negative cases, and we worked on this NGS system, what we have developed. And these are the subjects pathogens, some pathogens we have detected in the known etiology. I mean, you should have positive control and you should have negative control. That's it. That is the first thing you have to test with NGS. And then we have done, I'm showing here some of the data because it is not published. It is just 50 patients we have submitted. But we have a data of around 400 patients we have done for NGS, where a PCR is negative and clinicians suspecting still a meningoencephalitis thing. So we worked on everything. MTB is the main thing which we have noted in our institute, most of the places. TB incidence is also very high in this part of the region. So TB, we found most of the things, and then other things are there. This is a filtered data. And then the interesting thing is, it was negative with the routine method, it was negative with the PCR, but NGS catch it. So this is the advantage of NGS. And you need a good team, good bioinformatician. You know, there are different pipeline, different things which you look into that thing. So we have to be very careful when we talk about NGS, because if you give it to, it's the same like if you give it to 4 places for CSF samples, you may get some different results. Give it to 4 bioinformatics and see the results. I mean, they'll come with different interpretation. So we have to be very careful with the NGS thing. It is not at the moment for routine diagnosis. Let me tell you very honestly, we worked a lot because there are a lot of disconcordance, there are a lot of issues, there are a lot of matching also. That's not. And the advantage at the moment, only I can say, it has detected cases which were negative by all means. So this is something important. If we can work on this NGS system, if we can have a kind of a targeted NGS system, and with more sensitivity and specificity, then definitely I'm working with a Illumina group that whether we can have a kind of a targeted panels which we usually see in our country, which we have seen in our previous data and all these things. So this is very important to work on these things. And there's so many things about NGS, maybe this thing. The other important thing is where unknown etiology cases where we didn't find anything. Maybe we'll also get these cases in our this project also. So Angel, I think it's possible we do get some cases where we don't find anything. Even NGS is negative. And it's not a small number, 30 to 40% cases where this is negative. So very important to see why we are not getting, even they have symptoms, even they have clinical symptoms, even they have some kind of this thing. So we are not able to find it out. So generally, when I discuss with all the CZID Institute, so there is one scientist to work on autoimmune angle, and he has shown that autoimmune might be the thing, because these are the cases may go into autoimmune thing where they have triggered kind of immune system triggered due to so many things, due to so many infections, especially GBS, MS. There are so many things we have seen might be the autoimmune cases and all these things. We are working on these things, and this is what exactly I have already talked about these things. And the challenges I have already indicated, QC failure in DNA samples, and then optimization of sample collection. As I have already indicated, you have to take care of even the environmental samples are there, skin flora is there. So handling procedure to improve sample quality is there. Discrepancy between NGS and routine diagnostics. This is going to be a common thing, and we are going to face it, because clinicians think in a different way and the NGS will give you answer in these things. So basically, we have to figure it out whether we need some kind of a designated bioinformatic pipelines or what, that we have to figure it out. So basically, targeted treatment strategy will be the best thing, I think, and customized molecular diagnostic assay will be the final thing. So we are working on it, and we are working with several groups. One is in IIT Mumbai. They have large proteomic system where we are looking into the proteomic angle of this issue, and working with ICGB to understand the immune atlas of this problem. So basically, let's see, molecular level, we have figured it out. We are working on it still, but we do have immune angle as well as proteomic signatures of the bugs and the host will be really important on these things. I don't know whether I discuss on all these things, but we do, since we are working on metagenomics, why I'm telling about CSF is the difficult samples to handle for metagenomics, because you have very low nucleic acid load in the sample. So that's the problem we face all the time. And the same thing we have worked with the breast milk metagenomics for working in the antibiotics for extraction method and metagenomics things we have worked out. The same thing we have seen in this aspect, and this we are using it for early exposure risk, for AMR genes and breast milk analysis. We are also working for the resistant bacteria, and basically just touching the area, how early life this can be, we can identify CNS infections from infants to severe CNS infections. The other thing which I want to add is the blood-brain barrier. I think I should not talk on it because my colleagues will be discussing on it. So maybe I'll be there. So maybe I'll add something in that meeting. Maybe the next, after 2 weeks, we have 3 weeks, 2 or 3 weeks, we have this BBB. And really, I'm working on it. And this is my favorite area at the moment. And these are the issues I feel we are working on it. And we worked and we have submitted in the IGMR also. There are different strains responsible for crossing the blood-brain barrier. So different answers, different host mechanism, different blood-brain barrier permeability. So limitation in the CNS therapy. So SIMS is working on these things because we have BBB model at our cell culture level. And we are trying to develop that BBB model in our institute so that we can understand everything about all these things. So last thing which I want to talk about is neuroinflammation. And you talk about infectious thing, you talk about neuroinfection thing, you talk about anything, but what we are forgetting at the moment, we are working at the moment in this laboratory, is neuroinfection thing. And this is what a summarized slide. We are looking at the pro and anti-inflammatory cytokines. Our papers published in the CSF research as well as in the cytokine journal also, and one is in the Lancet also, how the inflammation creates damage. Because generally we diagnose a case and then I'll give example of TBM. Just if you diagnose it and you start with anti-TB treatment, so they are going to kill the bugs, but the inflammation mediated things is not controlled by this thing. So we have done a follow-up samples and all these things. If you can see, we work on IL-2, IL-10. It's a short thing I'm telling you. We have huge data. We have all supplementary data with us, but I'm just telling you, we have seen when patients come to us for brain TB, we have seen pro-inflammatory cytokines is quite high in our cases. And when patient improve, those who are improved, the balance changes and IL-10 expressions is quite high, which is anti-inflammatory. So there is something going on in the microglial cell. This is an immunological angle which we have worked with the cell culture, with the clinical samples, with around 75 follow-up samples, 60 follow-up samples, where we have seen the changes in the cytokine profile. So I think we need to discuss about the pro and anti-inflammatory angle in a follow-up samples. And the patient who expired, I have data, the anti-inflammatory is never produced. And the pro-inflammatory is like anything. If you talk about IFN gamma, if you talk about IL-2, these are the two culprit cytokine characterized pathogens we have seen in these things. And not only this thing, even in the COVID, we have seen what damage we have with inflammation. So this lab is working on neuroinflammation as well as working on inflammation, whether it's in a non-infectious disease, obesity, or cardiac issues, or you talk about arthritis, you talk about any non-infectious disease. I personally believe the inflammation is the key where we have to take care of it. So this might be the different angle which we can see in neuroinfection cases and all these things. And when we talk about cytokine, the gut microbiome come into picture. So it's all linked. We worked on gut-brain axis also at the moment. And some of our clinicians trying to treat the gut to treat brain. So there are so many things are going on. Very difficult to come out with everything in 45 minutes. Just touching some of the areas. Believe me, there are so many things. We have 30 years of story here. So different things and different point we are working on it. So the same thing I'm telling you, you can see we are working on it. And I think microglial cells playing important role in this thing. And I've seen several papers where IL-10 is not only inhibit the production of this inflammation, but it also invades inflammatory cells, monocytes, lymphocytes, to terminate ongoing inflammation within the CSF, within the CNS. So I think to control immune-mediated damage, then switch over to autoimmune, switch over to AMR, neuroinfection, all are different angles of one disease. So this is what exactly we have shown in this thing. So I think we are working on one molecule as a therapeutic target, IL-10. So let's see how we have one project, DBT project, where we are taking care of this IL-10, whether this can be used a new molecule for this kind of problem. So that's what we are working on it. And lastly, in last 2 minutes, we engaged with one project from Chandipura virus. So a very good lab in ICGB. Anmol, Sujata, and Sam, and all these people we are clinical partner for them. And then we got initially very interesting results with the Chandipura. You know, the Chandipura originated is just 60 kilometers from here in a Bandara district. So first cases were reported from that side, and the name is given from a Chandipura village. So we have started our research from Chandipura, and we are trying to see whether, I mean, at the immunological level, if we can find it out, some antibodies which are reacting with this Chandipura antigen and have potential for therapeutic or diagnostic or something like that. So we do have some uh, interesting results. Again, it's a very early result, so I don't figure it out. We also got some Chandipura positivity in our institute also. So maybe we are using those samples also for isolation of those cases. And we have collaboration in Gujarat also, where recently we have Chandipura pandemic and a lot of children's die. So we have with collaboration with those group and collecting samples from this thing. So this will help us to understand the disease pathophysiology, pathogen also, and immune response. Because this ICTB is really a good place where they are working on it, and then this is we are trying to get some kind of a immune response, basically B cell receptor repertoire and human monoclonals to Chandipura virus. Ultimately, the idea is that so we should have monoclonal antibodies to this Chandipura. So just it's 5:15. So I think I don't have to tell you the more complicated thing is neuroinfection, um, high morbidity and diagnostic gaps also. And I talked about conventional diagnostics, how it affects diagnosis, zoonotic linkages. I talked about One Health, AMR, BBB, neuroimmune thing, inflammation, and there are other approaches which we are using at the moment. So just touching all those areas for your understanding, we have in-depth data, we have publications. The lab is having around 152 publications on different aspects of this thing. So, but still, my answer is same. We have understand little bit about the things which we have done, and we are applying it. The best part is even it's a 1%, we are applying it and we are using it for our patient, because ultimately the money we get for research is from income tax pay. So we have to return to them. That's the thing which we are working. So even we have started a science, scientist and society a community in our center also. And we are trying to translate our research in a simple language to society. This is important. So I can go to conference and explain scientists, they understand it. But how do the common men know about neuroinfections? How do the common men know about the AMR and all these things? So you have to tell them in their own language that you having 8 kg of bugs in your body. You are nothing other than bugs. So this is what we have to explain them. And then he understand how important are bugs, how diet is important, how blood-brain barrier is important, how climate is important. So this is the way you have to tell them. And then definitely they will understand, they will support us. They will come for us, supporting us, because getting samples, follow-up samples, consent for samples, unless you tell them that it is for you, and you have to be honest for that statement, unless you don't give them assurance that it is going to be work for you, nobody will support you. And that's the reason scientists are not very highlighted like all our actors and other celebrities, people, and our Gen Zs are fans of all these people. But the scientists who are working behind the curtain day and night, nobody knows him. Even my mother was not aware of what I was doing. So in the lab and all this thing. So this is all from my side. And this is just a kind of a overview, sensitization of different areas, not in-depth thing. Maybe we have 5 years, 3 years project. So maybe we can talk on specific topic, specific pathways and specific angles whenever there is a chance. And looking forward to everyone. I mean, looking forward all climate change team to our institute. Maybe if you say, we can host you and we can have one small workshop, conference, brainstorming or roadmap for next 5 years other than this project. So then definitely that can be submitted to ICMR for another thing. So thank you very much. And I'm just sharing picture when I was there in February CMC Vellore. Thanks to Angel for inviting me, and met with great people, humble people, but really, I mean, very intelligent people in CMC Vellore. So it was a really pleasure. Institute is good, people are good, and it's a pleasure. and it's a Dr. Tavri's memories with CMC Vellore. That's the reason we come into this project. That's the main reason we were coming to protect. And my people as Rima, Vedi, Amit, and Komal, and our clinician, those who are involved in these things, Neeraj Bhaithi, Dr. Dinesh Kabra, and Nitin Chandak. So this actually helping us a lot in this project, which we are working at the moment. So thank you very much for your attention, and happy to discuss with you if you have anything. Thank you so much.
Thank you, sir. That was a fantastic overview of all the work you've done. I think each of your slide could be a seminar on its own. Thank you for giving us an insight into the translational aspect of brain infections at your center. So I have a question. So, like, how do we, I mean, the challenges of incorporating the cytokine response in brain infections. In fact, that is one of my areas of interest. So how do we, because, to process the samples, it has to be done immediately. And, those are the challenges, because the more you leave it at, even in room temperature, we know, because ambient temperatures are so high that the cytokines just get denatured. So how do you go about this challenge? I mean, how do you sort that out?
No, we included in them in the routine because the shelf life actually you can do in a 3-4 days. That's not
Yeah.
a problem. I mean, within a week time, we
Okay.
have done some kind of analysis how the half life of the cytokines and all these things. So I think if we can do it within a week time, it's fine. So what we are trying to do, we do immediately. Like we
Okay.
have a different diagnostic team, not only research. So we do
Okay.
get clinical samples for routine thing and all these things. So we immediately do. One of my colleague will perform all this kind of a test.
Okay.
Yes, but if you store it and do it later on for any projects, I don't know whether it will give that kind of response.
Yeah, that's what. It's because the
Yeah.
denaturation is because
Yes.
that's the issue. So also even extreme temperatures, freezing it also will kill it. So that's one challenge.
It's
So
very interesting, Angel. When you work with the cytokines and all these things, you
yeah,
will find a different kind of a pattern and a very small cost thing. I mean, nothing to do with this thing. So you need a high setup and all these things. Kits are
yeah,
available, everything is available. And if you can have a small setup for a R&D basis, because I know one of your scientists, I forget his name. I was with him in CMC Vellore. So he showed me lab and all these things. So when he said first I'll start free and then I'll sell it. So
Mhm.
that's the sentence, because the same thing we have done for cytokines. Initially
Yeah,
we did it for research thing, and then people like it, and then they are paying it cost to cost things. So you have to start with something.
that's true. Okay. Sure, sir. So any questions from the audience? You can post it in the chat box. Okay, so far none. You can even mail our questions to climbindiaicmr@gmail.com. We will communicate with the concerned expert, sir, and then we will get back to you with the answers. So, thank you, sir. That was
Yeah.
a phenomenal talk. This talk will be available in our YouTube channel. I request all of you to subscribe to the YouTube channel as well. You can watch it at your own pace later. So, thank you all for attending this webinar, and good evening.
Yeah. Thank you, Angel. Thank you for providing us opportunity.
Thank you.